Hello friends, welcome to my channel Biofix Buddy. If you haven't yet subscribed, please do subscribe to my channel and put an end to all your doubts. Before we begin with the concept, we should understand the name of the chapter, that is molecular basis of inheritance. First of all, what does a molecule means? As we learned in the earlier classes, a molecule means smallest unit of a substance that is made up of one or more atoms bound together by chemical bonds. So you've got to know what a molecule is. It's the smallest unit of a substance that is made up of one or more atoms bound together by chemical bonds. So this molecular basis of inheritance, the second word, important word you see is the inheritance. Inheritance, as we learned in the chapter Principles of Inheritance and Variation, inheritance means the passing on of, it is the passing on of characters from the parents to offsprings. From parents to offsprings. So entirely this name, molecular basis of inheritance, means what is the basic unit of inheritance? The units which are responsible for inheritance. We have learned that physical unit of inheritance is the chromosomes. Now what are these chromosomes made up of is what we are going to learn in this chapter. Now if we move on to the introduction, what is the genetic material that is present in any organisms? Genetic materials means the chromosomes. Chromosomes are made up of something called DNA, which is the genetic material. Genetic material means which is responsible for genes and passing on of characters, that is inheritance. So many organisms have DNA as their genetic material, while some of them have RNA as their genetic material. What does DNA full form? Deoxyribonucleic acid. Nucleic acid. What is the full form of RNA then? It is ribonucleic acid. Now what is the difference between this DNA and RNA? Where does the difference arise is what we will look into. So what are these two, DNA and RNA, made up of? Again, they both are chain of nucleotides. They are chains of nucleotides. For example, one DNA strand is made up of multiple nucleotides. Nucleotide bound with another nucleotide. So it is a chain of nucleotides. And now you might arise with the question, what are these nucleotides and what are they made up of? A single molecule of nucleotide is made up of a sugar plus a nitrogenous base and a phosphate group. So this is called a nucleotide. A nucleotide is made up of sugar, nitrogenous base and phosphate. So the difference in the sugar makes the difference in RNA and DNA. The sugar is of two types. There are two types of sugars, that is ribose sugar and deoxyribose sugar. ribose sugar. So it is the difference in their structure that is making them ribose and deoxyribose. The name itself says deoxy means absence of oxygen. So there is absence of oxygen molecule at one place of its chemical structure. If we look into the chemical structure of ribose sugar, ribose are always pentose. Pentose means they have 5 sides. They have 5 sides. Now let's number those sides. 1, 2, 3, 4. This one has a oxygen group over here, so we are not going to count that one. 4. The places which have carbon are the one which we are going to count. This is 4 and this is 5. So at this second place, at this region, there is OH. So in ribose sugar, we have OH at the second place, but in deoxyribose, if we see the structure, it is also a pentose. Same to same it is. The structure is same. The same. Here there is an oxygen. The numbering is 1, 2, 3, 4, and this place is 5 over here. But at the second position, instead of having OH, it has only H. So that is the difference that forms this RNA and DNA. RNA has ribose sugar as its sugar. The DNA has deoxyribose as its sugar. And when we come to nitrogenous bases, there are two types of nitrogenous bases. Nitrogenous bases. Nitrogenous bases. There are of two types. Purines. Another one is called the pyrimidines. Pyrimidines. So out of the 3 molecules or 3 atoms that the nucleotide is made up of, we have learnt about sugar. There are 2 types of sugars, ribose and deoxyribose, which is the main difference forming substance in DNA and RNA. Next, nitrogenous bases, there are 2 types, purines and pyrimidines. These purines are adenine and guanine, and they are 2 ring structures. 2 ring structures with nitrogen at first place, third place, 7th place and 9th place. Okay, so these are purines. Adenine and guanine are purines, which are 2 ring structures with nitrogen at first place, third place, 7th place and 9th place. We'll draw a molecular, the chemical structure of it, and look how you'll understand this when I draw the chemical structure of this one. Next, if we see into the description of these pyrimidines, the 3 pyrimidines, we have only 2 purines, but there are 3 pyrimidines. Cytosine, the second one is thymine, and the third one is uracil. And one important thing you have to know here is thymine is present only in DNA and uracil is present only in RNA. A major point that you should remember and you should know is that the thymine, which is a pyrimidine, is present only in DNA. The uracil is present only in RNA. So these are single ring structure only. They have only 1 ring. And they have nitrogen at the first and third place. Now we'll see the structure of purine. C, N, let's put C over here, then another N, C over here, C over here, and let's bound it, close it. Now I told you it is two ring structure. We have drawn a single ring now, and another ring comes up from here, N. This one also has N over here. Then it is closed up, ending at C. Now this is not how you draw it. Still, we have double bonds at this place, at this place, at this place, and at this place. So these are double bonds over here. Now if we will number the places, numbering starts from here. 1, 2, 3, fourth place is here, fifth place is here, sixth place is here, seventh place is here, eighth place, and ninth place. As I explained you in the structure of purines, there are two ring structures. We have two rings, ring number 1, ring number 2, and nitrogen is present at first place, third place, then seventh place, and ninth place. This is an important question. Where nitrogen is present in a purine is it might be a question, so you should be sure and should be able to explain where the nitrogens are present in a purine. Next, if we look into the structure of pyrimidine, I told you it is a single ring structure. Let's take a C over here, bound it with another C on this side, then another C over here, then N over here, N, C, and it is closed. We've got one ring. It is a single ring only. We have double bonds at few places, here, here, and here. Now if we number it, we have to come from bottom. 1, 2, 3, 4, 5, and 6. So as I told in the description, it is a single ring structure and it consists of nitrogen at first place and third place. Clear? So the DNA and RNA are the genetic materials and they are made up of chain of nucleotides, and these nucleotides are made up of three substances, that is sugar, nitrogen base, and phosphate. The sugar is of two types. There are two types of sugars, that is ribose and deoxyribose. So that is the one which is making the difference in DNA and RNA. The DNA has deoxyribose as its sugar, while the RNA has ribose as its sugar. Then nitrogen bases are of two types again, the purines and pyrimidines. The purines are again two, adenine and guanine. These are the two purines present in the DNA and RNA. They are two ring structures with nitrogens at first place, third place, seventh place, and ninth place. In case of pyrimidines, there are three pyrimidines: cytosine, thymine, and uracil. Cytosine is common in DNA and RNA, but thymine is present only in DNA and uracil is present only in RNA. It is a single ring structure with nitrogen at first place and third place. Clear? And phosphate group, there are no types of phosphate group. It is a single phosphate only. It is a normal phosphate. So a bond of sugar and nitrogen, a bond of sugar plus nitrogen is called a nucleoside. There might arise a word called nucleoside. What is a nucleoside? It is a bond of sugar and nitrogen. And if a phosphate group is added to that nucleoside, then only will it become a nucleotide. So you should know the difference between nucleoside and nucleotide. Sugar plus nitrogen is equal to nucleoside. When you add a phosphate group to that nucleoside, it becomes a nucleotide. Now let's draw a full structure of a nucleotide. Structure like this. The sugar is a pentose. That means it is five-sided. 1, 2, 3, 4, 5 sides we have drawn. Let's number 1, 2, 3, 4, and 5. This has a O over here. So we have got the deoxyribose sugar over here. Next, we require a nitrogenous base. Where is the nitrogenous base attached? To the sugar. The sugar has a nitrogenous base attached to it at the first place itself. At the first place, a nitrogenous base is attached. Next, we have the phosphate group. Where is the phosphate group attached? The phosphate group is attached to the sugar at the fifth place. So, this is a nucleotide now. Nucleotide. A sugar, a nitrogenous base, a phosphate group. Altogether, it is a nucleotide now. So they have few names for its bonds. The bond between the phosphate and the sugar is called the phosphoester bond. It is called the phosphoester bond, and the bond between nitrogen and sugar is called N-glycosidic bond. Now let's go into detail of structure of DNA. The same thing, we know that DNA is made up of nucleotides only. We'll draw the same structure once again here. Pentose sugar. The difference is there is no oxygen group at the second place. That's all. Deoxyribose sugar. Let's number it. 1, 2, 3, 4 and 5. We have a phosphate group at the fifth place. And we have for example, let's take, I told you the different types of nitrogen bases are. Let's recall that one again. Adenine, guanine, cytosine, thymine. Uracil we can't consider because we are learning the structure of DNA now. We are learning the structure of DNA, and uracil is present only in RNA. So completely in DNA there are 4 types: A, G, C, T. Now let's take G as our nitrogen base over here. G means guanine. G means guanine. Again, I told you it's a chain of nucleotides. Obviously, we have to draw another nucleotide over here to make a chain of nucleotides to make a DNA. Let's draw another nucleotide here and let's bind them together. Let's bind the 2 nucleotides together. We have a phosphate group here at the 5th place, and let's take a nitrogenous base different. Instead of G, let's take T. Let's take T at this place. Now, how are these 2 nucleotides bound? There should be a connection between them. A bound should be between them. That is the bond that is formed between the 3rd place of the previous DNA and the phosphate group. This bond between one nucleotide to another nucleotide is called phosphodiester bond. The bond between one nucleotide to another nucleotide is called phosphodiester bond. Let's make the bonds again once more clear. The bond between the sugar and the phosphate is called phosphoester bond. A phosphoester bond. The bond between the sugar and the nitrogenous base is called the N-glycosidic bond. This is the N-glycosidic bond. Now we have always learned that DNA is a double-stranded structure. There are 2 strands. We have drawn 1 strand over here now. The opposite strand also should be drawn. But one point over here is it is in upside down position. The sugar is in upside down position. That means the ribose sugar should be drawn in this way. It should be drawn in this way. Carefully observe this one. I'll explain why it should be drawn upside down only. Draw the structure along with me. We have a phosphate group at the 5th place again. This is the first place, and it should have a nitrogenous base. This will be adenine. This one will be cytosine. So now from here you can see that there should be a bond between these two nitrogenous bases. A bond is always made to connect the two strands between the nitrogenous bases only, and these bonds between the nitrogenous bases are the hydrogen bonds. Hydrogen bonds. Why is this strand in upside down form is, just imagine if we draw this strand also same as this one, these nitrogenous bases would face outwards. It would come onto this side as it is on the if we have seen on this side. It would project out, so outwards the nitrogen bases. So when it is at that position, obviously the two nitrogen bonds of this strand and this strand cannot bind with each other, because the nitrogen bonds will be facing outward on this strand. At that time a bond cannot be formed. That's why, to project the nitrogen bases inwards, the sugar or the strand is in upside down form. So if they ask you to describe the structure, there would be a question in your examination asking describe the structure of DNA. First point you can see in the structure is the DNA is a chain of nucleotides, which is made up of deoxyribose sugar, nitrogenous base, and a phosphate group. The second point here is the nitrogenous base is attached to the sugar at first position via N-glycosidic bond. And the third point here is the phosphate is attached to the sugar at fifth position via the bond between the phosphate and sugar is called phosphoester bond, via phosphoester bond. The next point is two nucleotides through third position and fifth position of sugar forms a chain of nucleotides by phosphodiester bonds. The two strands of DNA are bound by hydrogen bonds between the nitrogenous bases of opposite strands. So this is a clear explanation of the structure of DNA. We have two strands of DNA over here, and it is a chain of nucleotides. The first point is a DNA is a chain of nucleotides which is made up of deoxyribose sugar, nitrogenous base and a phosphate. Second point, in nitrogenous base is attached to the sugar at first position. This is what I am explaining now. At the first position via a N-glycosidic bond. The bond between the sugar and nitrogen is called the N-glycosidic bond. The phosphate is attached to the sugar at fifth position via phosphoester bond. So this is the bond I am explaining. It is the phosphate is attached at fifth position and the bond between them is called the phosphoester bond. The two nucleotides through third position and fifth position of the sugar form a chain of nucleotides. Now this is a fifth position of one nucleotide, this is the third position of one nucleotide, and they are making a bond between the two nucleotides. So the bond between them is called phosphodiester bond. They're attaching the two nucleotides, forming a chain. So the two nucleotides through third position and fifth position of the sugars form a chain of nucleotides by phosphodiester bond. And the last point is the two strands of DNA are bound by hydrogen bonds between the nitrogenous bases of the opposite strands. So now we have opposite strands and there is a bond between the nitrogenous bases, this one, and that is hydrogen bonds which are attaching the two strands and which makes the structure of DNA. I hope the structure of DNA is clear. Do they have any particular names that based on the nitrogenous base it consists of? Yes. Each nucleotide has a particular name based on the nitrogenous base it consists of. So, first one, if it has an adenine plus deoxyribose sugar, this will become a nucleoside, and the name of the nucleoside consisting of adenine as its nitrogen base is called deoxyadenosine. And if a phosphate group is added to it, it would form a nucleotide. So the nucleotide consisting of adenine as its nitrogen base is called deoxyadenylic acid, or another name of this one is deoxyadenosine monophosphate. The second one is, if it contains guanine as its and the sugar is same, deoxyribose, then it forms a nucleoside and it is named as deoxyguanosine. And if they add phosphate group to it, it will become a nucleotide and the name of it is deoxyguanylic acid. Another name for this one is deoxyguanosine monophosphate. The third nitrogenous base we have is thymine. If we have thymine as our nitrogenous base and the sugar is as it is, deoxyribose, it forms a nucleoside, deoxythymidine. What is it called? Thymidine. And if we add a phosphate group to it, it would form a nucleotide, deoxythymidylic acid. And another name of this one is deoxythymidine monophosphate. Now the fourth nitrogenous base that we have seen is cytosine. Plus we add the sugar, that is the D. So we add a deoxyribose sugar. Then it forms a nucleoside. And the name of this nucleoside consisting of cytosine as its nitrogenous base is deoxycytidine. So if we add a phosphate group to it, it would form a nucleotide which is called deoxycytidylic acid. Another name for this one is deoxycytidine monophosphate. So I hope the structure of DNA is clear. So let's have a look again through this. So when we have started the chapter, we understood what new molecular basis of inheritance is, what molecules are, and what we are going to learn in this chapter. When we come to the genetic material, obviously the genetic material we know is present in the nucleus, and the genetic material is called the chromosomes. Now chromosomes are made up of either of the two, DNA or RNA, right? So in majority of organisms, DNA is the genetic material, while in some lower organisms, the RNA is the genetic material. So both of them, DNA and RNA, are made up of nucleotides only, but the difference comes in the type of sugar it consists of. The DNA consists of deoxyribose sugar, while the RNA consists of ribose sugar. So the nucleotides are made up of sugar, nitrogen base, and phosphates. The sugar is of two types, ribose and deoxyribose. As I told, the DNA consists of deoxyribose and the RNA consists of ribose sugar. The nitrogenous bases again are of two types, purines and pyrimidines. The purines are adenine and guanine. Both are common in DNA and RNA. Then it is a two-ring structure. Purines are two-ring structures with nitrogens at first place, third place, seventh place, and ninth place. Then pyrimidines are single ring structures only. There are three pyrimidines: cytosine, thymine and uracil. The thymine is present only in DNA while uracil is present only in RNA, and the single ring structure has nitrogen at the first place and the third place only. And another point you should remember in the DNA structure is the purines are always bound with pyrimidines. Purines are always bound with pyrimidines. So if this structure, on one side if there is a pyrimidine, there should be a purine on the opposite side strand. So always, now you can see here the adenine is a purine and thymine is a pyrimidine. In this structure you can see adenine and thymine are bound. Okay. Similarly, guanine is a purine. C, that is cytosine, is a pyrimidine. So they are only bound. In opposite strands there should be purine and pyrimidines, which are going to be bound by the hydrogen bonds. Clear with that point? The next, if we see the structure of an entire nucleotide, we have a pentose sugar, that is the deoxyribose. If we come into if we see RNA, it is a ribose sugar. If we see a deoxyribose sugar, that is DNA. So we have a pentose sugar and phosphate present at the fifth position of the sugar, and there is a nitrogen base attached at the first position of the sugar. The bond between this phosphate and the sugar is known as phosphoester bond, and the bond between the nitrogen base and the sugar is called N-glycosidic bond. And as I told, RNA or DNA is a chain of nucleotides. Obviously, the nucleotides should form a bond between themselves. So, if one nucleotide is bound with another nucleotide, the bond between them is called phosphodiester bond, which I have mentioned over here. And that phosphodiester bond is formed by the third position of one nucleotide to the fifth position of one nucleotide. Based on the nitrogen base that nucleotide consists of, they are of different names. If the nucleotide consists of adenine as its nitrogen base, if we add a deoxyribose sugar, it forms a nucleoside. A nitrogen base plus sugar is equal to nucleoside. If phosphate is added to that nucleoside, it becomes a nucleotide. Simple formula. Then if it consists adenine as its nitrogen base, the nucleoside is called deoxyadenosine, and if we add a phosphate group to that deoxyadenosine, it becomes a deoxyadenylic acid, or it is called deoxyadenosine monophosphate. If it has guanine as its nitrogen base, the sugar deoxyribose, then the nucleoside it forms is deoxyguanosine, and if we add a phosphate group to it, it is called deoxyguanosine monophosphate. Similarly, if thymine is the nitrogen base, the nucleoside is called the deoxythymidine, and if we add a phosphate to this nucleoside, it becomes a nucleotide which is called deoxythymidyllic acid or deoxythymidine monophosphate. The next one, cytosine, is the nitrogen base. Then the nucleoside which is formed is called deoxycytidine, and if we add a phosphate group to the deoxycytidine, it forms a nucleotide which is called deoxycytidylic acid.